Hi, in this video we'll be talking about the structure of DNA and RNA. When we talk about the structure of DNA and RNA of prokaryotes and eukaryotes, RNA is generally single-stranded as we see here, while DNA is generally double-stranded. The common feature between RNA and DNA is both of them have a backbone which is made up of sugar molecules. So when I say sugar molecules, I'm talking about ribose sugar. Ribose sugar. Ribose sugar is a pentose sugar, that is, it is made up of 5 carbon atoms. So it is a 5 carbon ring structure. So these pentose sugar makes backbone, and then we have these nitrogenous bases. These nitrogenous bases are also called as nucleobases because they form the basic structure of the nucleic acids. Other than that, we have phosphate bonds which are not clear in this diagram. We'll be looking at those in the next diagram. Phosphate bonds, they connect the sugar molecules in the backbone, and we have hydrogen bonds. Hydrogen bonds, they connect the nucleobases of the two strands together in DNA molecule. So with this basic, we'll move on to the next slide. In this slide, we can see the picture more clearly. So what we can see here is the bases. So you can see these ring structure, these are bases. Then here we can see the sugar, that is ribose sugar. And here you can see the phosphate bonds. So these phosphate bonds are linking one sugar molecule with the next sugar molecule. And ultimately what we get here is a backbone. So this is the backbone. So if I draw, this will be the backbone. And if you look at this side, these are the nucleobases. So when we actually draw a DNA, so we kind of draw nucleobases projecting from this backbone of sugar molecules which are connected by phosphate groups. Now this is a single strand. When we talk about a DNA, we talk about another strand. So I have this another strand, and I'll draw the bases in pink color for this strand. And what happens is these bases of one strand are linked together with the next strand by hydrogen bonds. So these are hydrogen bonds. We can see here more clearly. So to revise what we have the sugar molecules connected by phosphate bonds, and then we have nucleobases connected by hydrogen bond. Now in the next slide, we'll be talking about the nomenclature, what a particular group of these molecules is called as. So let's go to the next slide, which is a little bit more detailed version of this slide. So it is more close up. So as we saw in the last slide, we had been talking about sugar. So this is the sugar here. And then we have the nitrogenous base or the nucleobase here. So in this diagram, we have two nitrogenous bases. This is one strand of one side, and this is another strand of another side. So we're looking at a DNA molecule, and this is the phosphate bond which will be linking it to the next pentose sugar, which is not seen in this diagram. Now if we take all of them together, that is on one strand, if we take one nitrogenous base, one sugar plus one phosphate, we call it nucleotide. But if I just exclude the phosphate part and I just consider sugar and a nitrogenous base together, this part is called as nucleoside. So that is the difference between nucleoside and nucleotide. That's just the terminology for the various grouping. So nucleotide is a basic backbone structure. So what is a DNA? DNA is nothing but a polymer of nucleotides. So we can say many nucleotides come together, join, and they form one strand of a DNA, which is linked to the next side strand with hydrogen bonds here. So these hydrogen bonds are linking together nucleobases of one strand to the nucleobases of another strand. So ultimately they are linking together the strands. Now as we saw the nomenclature, so we have various types of nucleobases. The most which are the backbones of the nucleic acids are adenine, guanine, cytosine, uracil, and thymine. And when we talk these bases in terms of nucleoside, that is bases plus ribose sugar, the terminology is, they're called as adenosine, guanosine, cytidine, uridine, and thymidine. So look at this, don't confuse with cytosine. Cytosine is a base while nucleoside version of it is cytidine. Now when we talk about a nucleotide version, that is, it contains a phosphate group attached to it, it is called as adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, uridine monophosphate, and thymidine monophosphate. So hopefully that should be clear. This is nitrogenous bases. When we add sugar to it, it becomes nucleoside, and when we add a phosphate to it, it becomes nucleotide. Now these nitrogenous bases, they come under two groups, that is purines and pyrimidines. Now purines are bi-ringed or double-ringed structure. So if you look at these nitrogenous bases, you can see they have nitrogens. Obviously that is the reason they are called as nitrogenous bases, and they have carbon and hydrogen, and they can have two rings or one rings. The two ring structures are called as purines, while the single ring structure called as pyrimidines. So we have adenine and guanine under purines, adenine and guanine under purines, and then we have pyrimidines here, pyrimidines. In that we have uracil, thymine, and cytosine. So there is one more rule that we notice in DNA is a purine always pairs with a pyrimidine. So we do not see a purine pairing with a purine, we do not see a pyrimidine pairing with a pyrimidine, but they have these different group bases. They pair together with hydrogen bonds when the two strands of DNA come together. So this is one more thing. And when we talk about the RNA and DNA strand, there are two differences in RNA and DNA strands. So one difference is that DNA consists of these four base pairs, AG and TC. So these are the base pairs in DNA. So it does not have uracil. So uracil is basically in RNA, which does not have thymine. So in RNA, uracil replaces thymine. So that is one of the important difference between RNA and DNA, other than the first difference that we previously saw, that RNA is single stranded. One more difference that we see in RNA and DNA is RNA consists of ribose sugar, while DNA consists of deoxyribose sugar. Let's see in the next slide what does that mean. Now look at this. This is a sugar structure. Both RNA and DNA have this 5-ringed sugar structure, which is a ribose sugar. But when we have this ribose sugar in RNA, it is a total ribose sugar. But when we have the same sugar in DNA, it has one of this oxygen missing. This oxygen is missing. That is the reason it is called as deoxyribose sugar in DNA. Other than that, we already saw we have purines and pyrimidines, and we have the phosphate bond that joins the sugar molecules. There's one more thing that we have to notice, that this phosphate bond joins 5th carbon of one ribose sugar to the 3rd carbon of the 2nd ribose sugar. So if we have this ribose sugar, if we have the 5th carbon here, it will join the 3rd carbon of the next, and then we have a 5th here, and then it will join the 3rd carbon of the next sugar, and that way. So when we read this strand, we read it from 5 towards 3 directions. So we read it 5' to 3'. So till now what we saw is, let's just summarize it. So what we saw is DNA is made up of 2 strands. Every strand has a backbone made up of deoxyribose sugar in DNA, Every deoxyribose sugar is connected to the next deoxyribose sugar by a phosphate bond. So we have phosphate bond. Phosphate bonds links 5 carbon of one deoxyribose sugar to the 3 carbon of the next deoxyribose sugar. So if we consider this strand, probably we'll call it 5-3- strand, and if we consider the other strand, it will be in this direction. It will be running in this direction. Now when we talk about these structures, these are nitrogenous bases which are attached to the sugar molecules, and nitrogenous bases are attached to the nitrogenous bases of the opposite side strand with hydrogen bonds. So these are hydrogen bonds here. So that's the basic structure of a DNA. Other than that, once we have a basic DNA, DNA is not stored in such uncoiled manner in our nucleus. It is stored as chromatids. Now what is chromatid? Chromatid is nothing but a supercoiling of DNA, and the enzyme that is used in this supercoiling, that drives the supercoiling, is called as topoisomerase. This is the enzyme. Now if you notice, this is a DNA strand which is coiling, but we need something else for it to coil. So these rounded structure, they are another protein structure which are called as histones. They are the protein structure. They provide kind of support for coiling. And if we notice, these coiling, when it is approximately 10 nanometer diameter, it is approximately 10 nanometer diameter, and approximately 2 or 3 coils together called as nucleosome, and a bigger section of it, a group of nucleosomes, is called as chromatid. And further chromatids are again supercoiled, and then it is again supercoiled, and this is how the DNA is packaged into such a small nucleus. So this was about the structure of DNA. And there's one more term that we need to understand. Whenever a DNA is loosely packed, it is the loose packing. Whenever there is loose packing, it is called as euchromatin. So euchromatin is loose packing of DNA. But when it is densely packed, here, it is called as heterochromatin. So the things that you should know in a structure of DNA is about the ribose and the deoxyribose sugar, how a sugar is connected to the next sugar, that is by a phosphate bond, then how nitrogenous bases are attached to the sugar to form nucleoside, and nucleoside along with a phosphate bond is called as nucleotide, and this whole nucleotide repeated many times makes one strand of a DNA or an RNA. In a DNA, one strand is linked to the other strand with hydrogen bond between the adjacent nitrogen bases. One more thing that we need to know is a pyrimidine type of a nitrogen base always pairs with a purine type of nitrogen base. Other than that, what we need to know is about the chromatin condensation. That is how the DNA coils over histones with the use of an enzyme topoisomerase to form chromatids, and how supercoiling of the DNA occurs, and how the loosely coiled structure of DNA is called as euchromatin, while the heavily coiled structure is called as heterochromatin. So that was a summary of the structure of nucleic acid. Thank you.